Today we are going to discuss about Gish. It is one of the important bioinformatic techniques. So in this Gish technique, we are going to discuss about what is Gish and principles of Gish and procedure of Gish and their applications in genomic studies. So Gish is shortly abbreviated as genomic in situ hybridization. So genomic in situ hybridization, that is Gish, is one of the molecular biological techniques which is used to detect the DNA sequencings within the chromosomes or within the living cells. It can be one of the most powerful technique or tool for studying the presence of specific molecules within the cells. So what are the principles behind the Gish? So first of all, the Gish techniques is based working under the principles of hybridization technique. So in which hybridization technique, the labeled DNA probe molecules can be used. So these DNA probe molecules is labeled with the help of fluorescent dyes or by using radioactive elements. These hybridized or labeled DNA probe molecules can easily or specifically bind to the complementary sequences in the target DNA within the chromosomes or even the cells. These cells are considered as our target cells. Thus, this technique makes use of in situ hybridization, which means these techniques are used to detect the molecular sequences occur within their natural locations inside the cells, rather than the extracted or then isolated from the cells. So during this technique, we should find the we can easily find the location of cells inside their chromosome lines or their sequences only. We don't need not to extract or isolated from the cells or from anywhere. In Gish techniques, the DNA probe which is complementary to the target region, after that which is used to hybridized within the DNA, within the chromosomes. So these techniques visualization can be done by using the fluorescent microscopic rather than imaging techniques. So what are all the procedures behind Gish technique? So first of all, we should prepare sample preparation. For the sample preparation, we need cells or chromosomes for a particular target genomes which was collected and prepared on the slide only. And the next step is denaturation. During the denaturation, we all know the DNA cells is denatured with the help of heating or by using any other chemical denaturant. The third step is probe preparations. So probe means the probe contain only oligonucleotide. The oligo means the genes contain the base pairs contain more 1 to 10 base pairs only genes. That is complementary to the target. Which these DNA probe should be labeled with a detectable marker, that is fluorescent or any other radioactive elements. The fourth step is hybridization. The labeled probe is applied to the denatured chromosomes or the cells. So these are all the basic and basic steps for procedure of Gish. First one is sample preparation, then second one is denaturation, fourth one is probe preparations, third one is probe preparations, fourth one is hybridization. So after hybridization, we are just wash with the water. The unprobed or unhybridization element should be washed out with the help of water. After that hybridization done, we should detect the content or a particular particle with the help of fluorescent microscopic techniques. And then final one is analysis. So analysis is typically by observing the location of the probe signal under a microscope, the presence or absence of their position, and with the help along with their probe signal can provide the insight value of the molecular content of the cells. So these are all the diagrammatic representation of Gish technique. So first we take a slides and then first we take a target cells, then take a slides and then apply a heat under the slide to get the denatured DNA molecules So after that probe hybridization will done. After that after hybridization we can easily deduct and analyze the molecule for exact detecting of our chromosome numbers or genes or any others. So these are all applications of Gish. With the help of Gish techniques, we can easily make a chromosome mapping for our future research work. And then second one is to detect the specific DNA sequences within the chromosomes or within the living cells along their genome sequences part. So third one is plant and animal genomics. Fourth one is studying the chromosomal rearrangement. So final one is cancer research and microbial genomics studies. Thank you.